DNA, deoxyribonucleic acid, is a molecule that carries the genetic information that determines the characteristics of all living organisms. DNA is composed of 4 nitrogenous bases, adenine, guanine, cytosine, and thymine, arranged in a double helix structure and connected by hydrogen bonds. The sequence of these bases determines the instructions for the development, functioning, and reproduction of an organism. DNA replication and protein synthesis are the two main processes through which DNA influences the biology of an organism. DNA is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. DNA evidence plays a crucial role in forensic science, particularly in criminal investigations. It can be used to identify individuals, connect individuals to crime scenes, and exclude individuals as suspects. DNA evidence is collected from various sources such as blood, saliva, semen, skin, hair, and other bodily fluids or tissues. It is analyzed in a laboratory using DNA profiling techniques such as polymerase chain reaction, PCR, and short tandem repeat, STR, analysis. The resulting DNA profile is then compared to reference samples, such as a suspect's DNA or a DNA database, to establish a match or exclusion. The high accuracy and reliability of DNA evidence make it a valuable tool in the criminal justice system. DNA evidence is widely used as a forensic tool in criminal investigations due to its uniqueness and high accuracy in identifying individuals. The following is a more detailed explanation of how DNA evidence is collected, analyzed, and used as evidence in forensic science. Collection. DNA evidence can be collected from a variety of sources including blood, saliva, semen, skin cells, hair, and other bodily fluids or tissues. Samples are usually collected at a crime scene, from a suspect, or from a victim. Proper collection and storage techniques must be used to ensure the integrity of the DNA sample. Analysis. DNA samples are then analyzed in a laboratory using DNA profiling techniques such as polymerase chain reaction, PCR, or short tandem repeat, STR, analysis. PCR is used to amplify specific regions of the DNA, while STR analysis involves identifying repeating patterns of DNA base pairs at specific locations on the genome. Comparison. The resulting DNA profile is then compared to reference samples, such as a suspect's DNA, a DNA database, or a known sample from a victim or crime scene. If a match is found, it can be used to establish a link between the suspect and the crime scene. If there is no match, the suspect can be excluded as a suspect. Interpretation. Interpretation of DNA evidence can be complex and requires specialized knowledge and expertise. Factors such as the type of sample, the size of the DNA sample, the presence of contaminants, and the quality of the DNA extraction and analysis can affect the interpretation of the results. Validity. The validity and reliability of DNA evidence depends on several factors, including the quality of the sample collection and analysis, the qualifications of the laboratory personnel, and the use of proper quality control procedures. DNA evidence must meet strict legal and scientific standards to be admissible in court. Overall, DNA evidence is considered to be one of the most powerful tools in forensic science, providing a high level of accuracy in identifying individuals and linking them to crime scenes. However, its validity and interpretation must be carefully considered to ensure that it is used appropriately and responsibly.